Hello, guys, and welcome to part two of the nervous system. So, in part one, we covered the parasympathetic and we covered the central nervous system and the peripheral nervous system. Now, we're gonna go into what the nervous system actually is. And it all starts with this one thing right here, which is called a neuron. This neuron has a very important job in the nervous system because it is what sends and receives signals or stimuli, and it basically makes us do the actions that we need to do. And that includes breathing, reflexes, digestion. Literally everything is controlled by this. This is like your brain. This is literally the brain of your body, the brain of your cells. So let's go into the anatomy of it first. So it all starts here in this section right here, which is called a dendrite. So a dendrite basically detects stimuli and sends the signal along the membrane to the cell body. And that takes us to the cell body. So the cell body is what makes proteins, and there's also a nucleus in the cell body as well. And then we go down to the axon. Now there's always a max 1 per neuron, and what this does is it sends signals towards other neurons, muscles, or glands. So it's how the neurons communicate and how the neurons communicate with each other and other muscles and glands. And then at the end here, we have our axon terminal, which is where the neuron communicates again with other neurons and other muscles and glands. And in the ends over here, you'll see these little pockets, and they include vesicles that have neurotransmitters. Neurotransmitters are just messengers that aid in the signaling between neurons, so from one neuron to another. So let's talk about all the stuff going on here, because this is very important, because this is how a signal is sent. So we have this spot right here, which is a gap. These gaps of the axon are called the node of Ranvier, and then these little white spots are the myelin sheath. The myelin sheath is basically just some fatty connective tissue that insulates and protects electrical signals, and they speed up the signal rates. So not every neuron has the myelin sheath. This is only included in some neurons. And do you remember how I said that it speeds up the signal, so it goes faster? So with the myelin sheath, we're talking about 150 meters per second. Now, not all neurons need to be going that fast. Like, for example, the neurons in your digestive system. You don't want to be digesting food at 150 meters per second. That's just not going to go well. So the neurons in your digestive tract, they're not going to have the myelin sheath, so they're going to go 5 meters per second. So the myelin sheath just speeds up, and it's not always on every neuron. Now, there's also these cells, these little hearts. They're called the Schwann cells. They are a glial cell, which is a support cell to the neuron. We're going to go into depth into that a little later. But it's basically a support cell, and it supports the neuron. So that is the anatomy of the neuron. It's pretty simple, pretty easy. There are 3 types of neurons. There is, the one that we're looking at right now is what we call the multipolar neuron, because it has the most extensions coming out of it. So we have all of these extensions that are coming out of the cell, so we call that multipolar, and this makes up all of the motor neurons and interneurons. It is most abundant in the body. So 99% of your neurons look like this. Do I have one? No, I don't. Okay. The other ones, they're going to look something like this. We're going to draw them. So we have bipolar, which is actually going to look like this. So you have the cell body, and then you have 1 extension and 2 extensions that are coming out of the cell body. So when we're talking about extensions, we're talking about the extensions that are coming out of the cell body specifically. So as you can see here, this is coming out of the cell body. This is coming out of the cell body. All of these are coming out of the cell body. When we're talking about a bipolar we are talking about only two extensions that are coming out of the cell body, and that includes this one and this one. It does not include these. So we're only talking about the extensions that are coming out of the cell body. So we have, so far, multipolar and bipolar. And the next one is going to be unipolar, which basically just means that it has one extension coming out of the cell body. So this is the cell body, and it can look something like this. So you have one extension, but it splits into two. But again, we're only talking about the extension that comes out of the cell body. In this case, we only have one extension. So that would make it unipolar. So we have multipolar, makes up all motor neurons and interneurons, and it's the most abundant. There's about 99% of your neurons are made up of this one. And then we have the bipolar one that has two extensions coming out of the cell body. And this one makes up special sense neurons, like the ones that are in your ear, eye, and nose. And then finally, the unipolar that has only one extension coming out of the neuron makes up general sense neurons, and this includes pressure, pain, and temperature. So, now we're going to talk about the glial cells. So, these cells, let's do this one in pink. So, these cells are called glial or neuroglia cells, and they are basically your neurons' helpers, and they outnumber neurons by a lot. So for every one neuron, there is 50 glia or neuroglia, and all they do is they support, protect, and speed up nerve impulses. Like they bring nutrients to your neurons to help them grow. So glial cells, they can repair themselves if damaged, but neurons, they cannot be repaired. So that's important. Glial cells, they can repair themselves, but neurons cannot repair themselves. Glial cells cannot generate nerve signals. Neurons generate nerve signals. So neurons, they generate nerve signals, but they cannot repair themselves. Glia do not generate nerve signals. They only support the neurons, and they can repair themselves if they're damaged. Now, the reason why neurons cannot, regenerate or repair themselves if damaged is because as they age, they lose the centrioles, and so they can't regenerate, they can't multiply. And an example of the glia cell, again, is going to be this Schwann cell right here. Alrighty, so how do neurons send signals? Okay, we're talking about how they always sending signals. How do they actually send these signals? There's something called the action potential, which is also called a nerve impulse or signal. And it is a localized electrical event that only occurs in the node of Ranvier. So the node of Ranvier is the gaps in between the myelin sheath. So this action potential only happens in these gaps, and it jumps from one gap to another gap until it reaches the axon terminal. So there is 3 stages to the action potential, and they are, I think I have them here, let's see. Okay, yes. Alright, let's do it down here. So there are 3 stages of action potential. So stages of action potential. The first stage is going to be depolarization. All of this is going to make sense in a second. What does polarization mean? What is this? What is the goal here? But depolarization has 2 important things, and that is the threshold and then propagation. And then the next stage is going to be repolarization. And then the final stage is hyperpolarization. Polarization. Now, before we even get into action potential and all of that stuff, we have to understand what does polar mean. So polar just means opposite charges. So when we have a neuron, we have what we call a resting neuron. So that is basically when a neuron is not sending a signal because it doesn't have any stimulus. It's just waiting. It's just sitting there waiting. So what happens is there's this extracellular fluid outside of the cell. So we're right here. This is the outside of the not outside the cell, sorry, outside of the neuron. Extracellular fluid. And there's a high amount of sodium out here. And in the inside, in the intracellular fluid, there is a high amount of potassium compared to the outside. So there's also a negatively charged, that's not shown on the picture, but there's a negatively charged protein that is only inside of the cell. So that means if we have a negatively charged inside and a positively charged outside, that creates polar, that they become polar, because you have a positive on the outside, negative on the inside. Those are opposite charges, and that is what polar means. So the overall charge outside of the neuron is positive compared to the inside one. This creates polarity and a potential to do work because these opposites want to attract. So one thing that we really need to remember is, hold on, where's Nagaps? We need to bring her here. Let's grab her. Ooh, why does she have a white background? That's okay. Alright, Nagaps, you can just sit right there. Alright, so the resting membrane potential. So resting, actually I'm just going to abbreviate it. It's the resting membrane potential is always at negative 70 millivolts. So that is the resting membrane potential. So that is when the cell, what's it called? The neuron is resting. So, how does this work? Like, how do you maintain this difference? Something called diffusion, which is what's going on here. So what this is a sodium-potassium pump. So, these ions will leak across the membrane, but we have these sodium-potassium pumps that are along the axon, and they are always working. And these are protein pumps that are activated by ATP. So this is our ATP right here. It binds to it, and then it opens. So the pumps actively move sodium in and potassium out to make the extracellular and intracellular fluid balanced again. So let's say we have some sodium ions that leaked inside. So now we have sodium on the inside and sodium on the outside. So we need to let out some potassium so we can equal out the charges. So the ATP is going to come in here and it's going to give a phosphate. So it gives its phosphate. And then 3 sodiums. So it goes in the ratio of 3 sodiums for every 2 potassiums. So 3 sodiums are going to come out. The pump is then going to change its shape. So it goes from this shape to this shape. And it lets the sodium out. The sodium that leaked in, it lets it out. And then the potassium on the outside binds in. Remember, 3 sodium out, 2 potassium in. So they come in here, and then the phosphate leaves and the cell shape goes back to normal. And now we have a balanced level of sodium to potassium. So that's the sodium-potassium pump. It's always working to make sure that the levels on the outside and the inside are balanced. So just to recap, you have your it starts out with some sodium leaking in. You don't want that sodium in there. So the protein gets a phosphate from ATP, and that changes its shape, and it becomes this V shape instead of this shape, and that lets out the sodium, and then whatever potassium leaked out goes into this protein, and then the phosphate deattaches. So this phosphate is going to deattach, and that's going to change the shape of the protein, and the potassium is going to go back into the intracellular fluid. So that is the sodium potassium pump, and it keeps doing that to maintain this. Now, there is something called the voltage gated channels, and these are channels that open when there is a change in ion levels. What that means is when there is a signal, they are going to open. So a change in ion levels occurs when a signal from the cell body reaches the beginning of the axon. So from here, when it reaches this beginning right here, that is when the ion levels are going to change, which are going to cause these gates, these voltage gates, to open. And there's two important types of voltage gates. We have the Na voltage gates, and then the potassium or K voltage gates. So let me just grab Nogops. Let me grab her with her seatbelt and everything. And we are going to get into the stages of the action potential right here. So, voltage gated channels. They only open and close when there is a change in voltage electricity. A change in voltage electricity occurs when a signal from the cell body reaches the beginning of the axon. So, how do these gates work? So at rest, at the resting potential, voltage gates are closed. So let's say this is the sodium voltage gate, and it's at resting. So right here, it's at resting, so it's closed. It's resting. It doesn't need to let the sodium in. So this sodium just doesn't go in. And then when any changes happen, the gate opens, and the sodium rushes. It rushes down into the negative area because it's polar. And that is how these gates work. So the same thing happens with potassium. The potassium gate is closed. When it opens, the potassium rushes out the other side. But we'll get more into that. So what exactly starts this action potential? So we know it starts at the axon entry, but what starts it? It's something called a grade potential. A grade potential is a stimulus that is picked up as an electrical or chemical change by the dendrites. So these dendrites, they pick up a signal, and it's picked up as a electrical or chemical change. And we know what happens when an electrical or chemical change happens. These voltage gates are going to open up. So the graded potential happens here, and then the action potential happens here, and then synaptic activity, which we'll talk about later, happens here. So now we're talking about the action potential. So we have our three stages that were down here. We are going to start with depolarization. So depolarization is all about sodium. So when you think depolarization, it's always going to be about sodium. So I'm just going to put it here, sodium. It's always sodium with depolarization. So the local electrical impulse travels from the dendrites. They're going to travel from in here into the axon. And the electrical impulse, this electrical impulse is going to alter the resting change in the intracellular fluid. So that means that there's going to be a change in charge. And so what's going to happen to the sodium gates? The sodium gates are going to open, and the sodium is going to rush in there. Inside, so now the sodium is inside of the neurons, this becomes less negative. So do you remember what the threshold was? We were at negative 70 millivolts. And when the sodium goes into here, this area that was negative 70 is going to become more positive. And it's going to become, from negative 70, it's going to go up to negative 55 millivolts, which is the threshold. Threshold. So remember, in depolarization, we have two main stages. That's threshold. So the sodium is going to rush in. The inside of the intracellular fluid is going to become less negative, so it's going to depolarize. That's why it's called depolarization. And what happens is that as the sodium goes down, like, see this first gate, the sodium is going to go down. So there's going to be a change in electrical charge here. So the next gate is going to open. And then the next gate is going to open. And then it's just going to keep going down until it goes down the entire thing. So it bounces from the myelin sheath until it goes into the axon terminal. So that's just going to keep happening down along the line. And the threshold is also called the point of no return, which means that all sodium gates will open and an electrical signal will travel down the entire axon. So remember how I said that it's like a domino effect? When one gate opens, it changes the charge of the other one, and then all the gates are going to open like a domino effect? That is what we call propagation. So threshold is when negative 50 millivolts is reached, and then propagation, we're just going to say domino effect of like all the gates opening. Alright, so now what do? All the gates are open and we've reached this state. So now we're going to go to repolarization. So what happens in repolarization, which is our second state? This repolarization is all about the potassium. So we said that the depolarization is all about sodium. Now repolarization is all about the potassium. And what the goal here is, now that the signal has traveled through, we need to go back to our resting potential, which was negative 70 millivolts. So we need to go back to this resting potential. How are we going to do that? We simply just open the potassium channels so that they even out. So if we have too much sodium inside, we have to let the potassium out so we can equal it out. So at the initial node of Ranvier, the intracellular fluid is positive down here. But then the voltage change causes the sodium gates to close, right? So these sodium gates are closed. Sodium can't come in anymore. And it opens the potassium gates. So now this potassium is going to leave the intracellular fluid and it's going to go to the extracellular fluid. And the charge in the intracellular fluid is going to slowly return to negative 70. So it has to go back to the resting potential, or negative 70, in order for a new signal to start. It cannot start a new signal unless it goes back to the resting potential. Now, there's one more little stage, and it's called the hyperpolarization. And basically, this hyperpolarization, what it is when the gates, like, let's do this in a little graph. So here we have negative 70 millivolts resting potential, and then we get a stimulus, and then we start heading towards this threshold, so that's negative 55 millivolts, and then here at the very peak is going to be our action potential, right? That's where all the Na gates open, right, open, and we have reached what this would be depolarization. This is depolarization. And then we're going to start going down, right? We want to be back at negative 70. So the Na gates are going to close. So Na gates close. And the potassium gates are going to open. And so we're going down here. But the thing about the potassium gates is they are slower at closing than the sodium ones. So they actually let out more potassium than needs to be there, like more potassium out than it needs to be there, because it has like a delayed period. They're slower at closing their gates. So we're gonna have this like bottom one where it's gonna dip to like negative 75. And then we have something called a refractory. Hold on, let me write it down here. Refractory period. And what the refractory period is, it's just a period that ensures that the action potential moves down the axon in one direction. So it just makes sure that it's all leaving, it's all going, and we're all chilling. So since the K channels have a slight delay when closing, more potassium is going to leave than required to make it to polarize the membrane. So it becomes more negatively charged than it was at resting rate, which makes the membrane hyperpolarized. That's why this stage is called hyperpolarization. And so if the cell is far, like it's way more far than it's way more far away from the threshold than it was before, it cannot generate an action potential. So that's why it needs this refractory period that will let it go back up. And what makes it go back up to the negative 70 that we want right here? It's going to be these sodium-potassium pumps. So we go back to these sodium-potassium pumps that bring us back to negative 70 millivolts. And that is basically how your cell your neuron sends a signal. So 3 stages. Depolarization, all about sodium. We have threshold and propagation. Threshold, we have to reach negative 55. Oops, what is this? Negative 55 millivolts. And then, okay, we're just going to ignore that. And then propagation, which is the domino effect. And then we have repolarization, all about the sodium going back out to make it polar again. And then hyperpolarization is that small period where it dips a little bit because the potassium channels are a little slow at closing. And then it comes back up to the resting potential, which is negative 70 millivolts. So that is how a neuron sends a signal. Thanks, guys. Did you learn, Nagabs? Did you get that? She really needed that seatbelt. This was a heavy one. But yeah, thank you, guys.